Hello. Ready to dive into another crucial topic today? I think understanding body fluids and circulation is so important, not just for class 11th and 12th boards, but especially for cracking NEAT, right?
Absolutely. Welcome, everyone. Yes, today we're tackling body fluids and circulation. It's fundamental, not just for exams, but honestly, for understanding how you work.
It feels like the body's internal transport system.
That's a perfect way to think about it. It's the lifeline. See, the human body is this incredibly complex machine, right? And every single part, down to the tiniest cell, needs a constant supply of essentials and, just as importantly, needs its waste removed continuously.
So how does the body manage all that delivery and pick up across different parts?
Well, that's where specialized transport systems come in. These rely on specific fluids moving through dedicated pathways. Our mission today, then, is to really unpack this. We'll get a thorough understanding of blood, lymph, the heart, the central pump, and how circulation works. We'll break down all the complex bits into digestible steps, ready for your exams.
Sounds great. So where do we start? The basic need for it all.
Exactly. Let's start with the why. At its very core, biology tells us that every living cell needs resources. Think nutrients, oxygen, other essential bits. And at the same time, metabolic processes produce waste. Carbon dioxide, other byproducts. These can be harmful if they just, hang around.
So it's a two-way street. Supplies in, waste out for every cell.
Precisely. Now, for simple organisms, maybe sponges or coagulants floating in water, it's relatively straightforward. They can just circulate water from their surroundings through their body cavities. That direct exchange works for them. Simple diffusion, basically.
But for complex organisms like us, that wouldn't work, right? Our cells are too far from the outside environment.
Exactly. Imagine trying to get oxygen to your brain cells just by circulating external water. Impossible. So evolution came up with a much more sophisticated solution. Specialized internal fluids. Fluids contained within the body, designed for efficient transport. In higher organisms, including humans, the main fluid, the most common one, is, of course, blood.
Okay, blood. But you mentioned another one, too.
Yes. There's also lymph. It plays a really important supporting role, especially in immunity and fat transport, as we'll see. So our roadmap for this deep dive is clear. First, we'll dissect the composition of blood and lymph, understand what they're made of. Then we'll get into the mechanics, the actual mechanism of blood circulation itself. How does it all move?
Got it. Starting with blood, then.
Let's do it. Blood. It's often called the lifeline fluid, and it's technically classified as a special connective tissue.
Connective tissue. Why connective?
Because it literally connects different parts of the body by transporting substances between them. It's made up of two main components. There's the fluid matrix, which we call plasma, and then the cellular bits, collectively known as the formed elements.
Okay, plasma and formed elements. Plasma first.
Plasma first. It makes up about 55% of your total blood volume. It's the liquid part. Appearance-wise, it's typically straw-colored and a bit viscous, slightly thick. Composition, mostly water. About 90 to 92%. This makes it a fantastic solvent, carrying everything along.
And the other 8–10%?
That's mainly proteins, about 6 to 8%. And these aren't just filler proteins. They have very specific vital jobs. Let's look at the main ones. First up, fibrinogen. Absolutely key for blood clotting or coagulation. When you get a cut, fibrinogen helps form the plug, your internal patch kit.
Okay, fibrinogen for clotting. What else?
Then you have globulins. Think of these as your body's defense force. They're heavily involved in immunity, fighting off invaders like bacteria and viruses.
Globulins for defense. Got it. And the third type?
Albumins. These are crucial for maintaining osmotic balance. They help keep the right amount of water in your blood vessels, preventing tissues from swelling up or getting dehydrated. They regulate fluid distribution.
Fibrinogen, globulins, albumins. Clotting, defense, fluid balance. That makes sense.
Right. And besides these big three proteins, plasma carries other things too. Small amounts of minerals, ions like sodium, calcium, magnesium, bicarbonate, chloride, all critical for various functions like nerve signals and muscle work.
Minerals too. What about nutrients?
Absolutely.
Right.
Nutrients in transit, glucose, amino acids, lipids absorbed from your food, traveling in the plasma to reach the cells that need them. And interestingly, those clotting factors we mentioned, they're also present in plasma, but usually in an inactive form, just waiting for the signal, like an injury.
Always ready, just in case.
Exactly. Now, a quick terminology point, very important for exams. Serum, Have you heard of that?
Serum, is that related to plasma?
It is. Serum is essentially plasma, but without the clotting factors.
Okay, so if I have a blood sample and I manage to remove all the red blood cells, white blood cells, and platelets, and then I also take out those clotting factors you just mentioned, like fibrinogen, what's left over is what we call serum, right? Is that how I should visualize it?
That's a very clear way to put it, and you've got the core idea there.
Yeah.
Precisely. Serum is what's left after the blood has clotted, and the clotting factors, especially fibrinogen, have been used up or removed. It's a key distinction, often tested, well identified.
Got it. Plasma minus clotting factors equals serum.
Okay, so what about the other part of blood, the formed elements?
Right, the formed elements. These are the cellular components. They make up the remaining 45% or so of your blood volume. This includes erythrocytes, leukocytes, and platelets. Let's tackle them one by one.
Erythrocytes first. Those are the red blood cells, RBCs.
Exactly. Erythrocytes or RBCs. They are incredibly abundant. Think about this. In just one tiny cubic millimeter blood, a healthy adult male has about 5 to 5.5 million RBCs.
Wow, that's a huge number. Where are they made?
In adults, they're produced primarily in the red bone marrow. And they have some really unique features. They have that distinctive biconcave shape, like a flattened disc indented on both sides. This gives them a huge surface area for gas exchange and lets them squeeze through tiny capillaries.
And they don't have a nucleus, right? At least in mammals.
Correct. Mature mammalian RBCs are devoid of a nucleus. This is an amazing adaptation. It frees up internal space so they can pack in more of their most crucial component, hemoglobin. Hemoglobin, that's the red pigment, contains iron.
That's the one. Hemoglobin is this red-colored, iron-containing complex protein. It's what makes blood red. A healthy person has about 12 to 16 grams of hemoglobin per 100 milliliters blood, and its function is absolutely critical. Hemoglobin plays a massive role in transporting respiratory gases, mainly carrying oxygen from your lungs to your tissues, and also helping carry some carbon dioxide back.
So they're like tiny oxygen delivery trucks. How long do they last?
They have an average lifespan of about 120 days. After that, they get old and are removed.
Where do they go?
They're destroyed primarily in the spleen, which is why the spleen is often called the graveyard of RBCs. Definitely remember that for your exams.
Spleen, graveyard of RBCs. Got it. Okay, next formed element, leukocytes.
Yes, leukocytes or white blood cells, ABCs. Unlike RBCs, they're colorless, no hemoglobin. Key features. They do have a nucleus, and they're much less numerous than RBCs. Only about 6,000 to 8,000 per cubic millimeter. And their lifespan is generally quite short. Now, WBCs aren't all the same. We divide them into two main categories based on whether they have granules in their cytoplasm.
Granulocytes and agranulocytes.
Exactly. Let's break down the granulocytes first, step by step. First type, neutrophils. These are the most common WBCs, about 60–65% of the total. Their main job is phagocytosis. They engulf and destroy foreign organisms like bacteria. Your frontline soldiers?
Neutrophils, bacteria eaters.
Okay. Second, basophils. These are the least abundant, only 0.5 to 1%, but they're important. They secrete histamine, serotonin, heparin chemicals involved in inflammatory reactions. Think of them as sounding the alarm during injury or infection.
Basophils, inflammation signalers. Small numbers, big role.
Right. And the third granulocyte, eosinophils, about 2–3%. They help resist infections, particularly parasitic ones, and they're also heavily involved in allergic reactions. High eosinophils often points to allergies or parasites.
Eosinophils fight parasites involved in allergies. Got it. That covers granulocytes. What about agranulocytes?
Okay, granulocytes. They lack those obvious granules. Two main types here. First, monocytes. These make up about 6–8% of WBCs. Like neutrophils, they're also phagocytic. They eat foreign invaders. They can actually migrate into tissues and become even bigger eaters called macrophages, the heavy-duty cleanup crew.
Monocytes, also phagocytic, become macrophages. Okay.
And finally, lymphocytes. These account for about 20–25% of WBCs. They are the specialists. There are two major types, B lymphocytes and T lymphocytes. Both B cells and T cells are responsible for the specific adaptive immune responses of your body. They target particular pathogens and create immune memory.
That's a lot of different white blood cells, each with very specific roles. How do they all work together in the body's defense? Like, what's the main difference between how neutrophils and monocytes protect us versus what the lymphocytes do?
That's an excellent question. Really gets to the heart of their teamwork. Think of it like this. Neutrophils and monocytes are your general security guards, the first responders. They attack any intruder they find quick, non-specific defense, general cleanup.
Okay, the immediate response team.
Exactly. Lymphocytes, on the other hand, are like your highly trained special forces, your intelligence agency. They identify specific enemies, a particular virus strain, for example. Then they launch a targeted attack, and crucially, they remember that enemy. That's adaptive immunity, the basis for vaccines.
Okay. General immediate defense versus specific, targeted, memory-based immunity. That clarifies it nicely. A great division of labor.
Precisely. A truly brilliant system. Now, the last of the formed elements, platelets, also known as thrombocytes. These are interesting because they aren't actually complete cells. They're tiny fragments broken off from very large cells in the bone marrow called megakaryocytes.
So just cell fragments, what do they do?
They play a critical role. Blood normally contains a lot of them, roughly 1.5 lakh to 3.5 lakh per cubic millimeter. That's 150,000 to 350,000. Their primary function, they release substances that are essential for coagulation, or blood clotting. They're central to stopping bleeding.
So they initiate the clotting process.
They're key players in initiating and forming the clot. Clinically, if someone's platelet count drops too low a condition called thrombocytopenia, they can have serious clotting problems, leading to excessive bleeding. They're vital for plugging leaks in blood vessels.
Okay, so that covers plasma and all the formed elements, RBCs, WBCs, and platelets. Quite a complex fluid.
It really is. Now that we know what blood is made of, let's talk about blood groups. This is hugely important, both clinically for safe transfusions and definitely for your NEAT exam. While blood looks the same, there are crucial differences based on markers on the cells. The two most important systems for grouping are the ABO grouping and the RH grouping.
ABO first. I know there's AB, and O.
Correct. The ABO system is based on the presence or absence of two specific antigens on the surface of your RBCs. Antigens are like chemical name tags. We call them antigen A and antigen B. At the same time, your plasma contains natural antibodies, proteins that react against antigens you don't have. Let's map it out. This is critical for exams. Blood group A. You have antigen A on your RBCs. Your plasma has anti-B antibodies. You can donate to A, and AB you can receive from A and O.
Okay, A has A antigen, anti-B antibody. Receives A, O donates A.
AB.
Perfect.
Blood group B. You have antigen B on RBCs. Plasma has anti-A antibodies. Donate to B, AB, receive from B.
B has B antigen, anti-A antibody. Receives B, O donates B, AB. Makes sense.
Blood group AB. You have both antigen A and antigen B on your RBCs. Crucially, your plasma has no antibodies, either anti-A nor anti-B.
Both antigens, no antibodies. So who can they donate to?
Only other AB people. But because they lack antibodies, they can receive from everyone, A, B, AB, and O. This makes them the universal recipients. Remember that term?
Universal recipients AB.
Yeah.
Got it. And O.
Blood group O. These individuals have neither antigen A nor antigen B on their RBCs, but their plasma has both anti-A and anti-B antibodies.
No antigens, both antibodies. So they can donate to
everyone, A, B, AB, and O. Because their cells lack the A and B antigens, there's nothing for the recipient's antibodies to attack. This makes Group O the universal donors, another key term.
Universal donors O, but they can only receive from O, right? Because of their antibody.
Exactly right. They can only receive O blood. So you see why careful matching before a transfusion is absolutely vital. If you give someone the wrong blood type, their antibodies will attack the donor RBCs, causing clumping, destruction of cells. It can be fatal. Strict matching is non-negotiable.
Definitely clear why that's so critical. Now, what about the RH grouping?
Right, the RH grouping. This involves another antigen called the RH antigen. It was first found in rhesus monkeys, hence the RH. Most people, about 80% of humans, have this RH antigen on their RBC surface. They're called RH positive, RH plus V.
And the other 20%?
They lack the antigen, so they are RH negative, RHVV.
Okay, positive means have it, negative means don't. How does this affect transfusions?
Here's the key point. An Rh negative person doesn't naturally have anti-Rh antibodies. But if they are exposed to Rh positive blood, say through a transfusion, their body will start producing anti-Rh antibodies.
So the first exposure primes them, and a second exposure could be dangerous.
Exactly. That's why, for transfusions, the Rh group must also be matched, along with the ABO group. Giving Rh plus V blood to an RHV person can cause serious problems, especially after the first exposure.
Makes sense. Match both ABO and Rh.
Precisely. And this leads us to a very important special case involving Rh incompatibility, something often tested in meat, erythroblastosis fetalis. Sounds complicated, but let's break it down carefully.
Okay, erythroblastosis fetalis. What's the scenario?
Imagine an Rh negative mother who is pregnant with an Rh positive fetus. The baby inherits the Rh plus factor from the father. During the first pregnancy, the mother's and baby's blood are mostly separate thanks to the placenta, so usually no major issues. However, during delivery, as the placenta detaches, some of the baby's Rh plus fetal blood cells can mix into the mother's Rhv bloodstream.
Okay, so the mother gets exposed to the Rh antigen at delivery.
Correct. Her immune system sees this Rh antigen as foreign and starts producing anti-Rh antibodies. This doesn't harm the first baby who's already born.
But what happens in the next pregnancy?
If the subsequent fetus is also Rh positive, those anti-Rh antibodies the mother produced can now cross the placenta and enter the fetal bloodstream.
And attack the baby's red blood cells.
Exactly. The mother's antibodies destroy the fetal RBCs. This can cause severe anemia, jaundice, due to bilirubin buildup from destroyed RBCs. It can even be fatal for the fetus. That condition is erythroblastosis fetalis.
That sounds awful. Can it be prevented?
Yes, thankfully. There's a very effective prevention strategy. The Rh-negative mother is given an injection of preformed anti-Rh antibodies like RhoGAM, usually around 28 weeks of pregnancy, and Ardor shortly after delivering an Rh-positive baby.
Giving her antibodies, how does that help?
These injected antibodies quickly find and destroy any Rh plus fetal-to-fetals that might have entered the mother's circulation before her own immune system gets a chance to react and produce her own long-lasting antibodies.
So the preventative measure for erythroblastosis fetalis is essentially a passive immunization for the mother. It's like giving her ready-made antibodies to stop her own active immune system from reacting against the baby's Rh-positive blood. Is that the right way to think about it for the exam?
That's a sharp observation and a perfectly accurate way to connect the dots for your exam. You've precisely captured the essence of it. It's a prime example of passive immunity used clinically to prevent a severe immune reaction. Excellent connection. That level of understanding is exactly what's needed.
Great, that clarifies the Rh issue and erythroblastosis fetalis. Phew. Okay, what's next? Clotting.
Yes, let's move on to the fascinating process of coagulation of blood, or clotting. It's a vital defense mechanism. Think about when you get a cut. It bleeds for a bit, but then usually it stops. Right? That's coagulation at work.
To stop us from losing too much blood.
Exactly. It prevents excessive blood loss. You see that dark reddish-brown scum that forms? That's the clot, or coagulum. It's basically a meshwork of protein threads called fibrins, which trap blood cells, like RBCs and platelets, to form a solid plug.
Fibrin threads trapping cells. How does that mesh form? Is it complicated?
It is quite complex. It's a cascade process, like a series of dominoes falling. Let's go through it step by step so you can visualize it. Step 1, injury trigger. An injury or trauma happens. This immediately activates platelets at the site, making them release certain factors. The damaged tissue itself also releases factors. That's the initial signal.
Okay. Platelets and tissues send out alarm signals.
Step 2, thrombokinase formation. These initial factors kickstart a chain reaction, activating a series of inactive clotting factors already present in the plasma. It's a cascade of enzymatic reactions. The end result is the formation of an enzyme complex called thrombokinase.
So the signals lead to making thrombokinase.
Step 3, prothrombin to thrombin. Now, thrombokinase acts as an enzyme. Its job is to convert an inactive plasma protein called prothrombin into its active form, thrombin.
Thrombokinase activates prothrombin into thrombin.
Got it. Step 4, fibrinogen to fibrin. Active thrombin now takes charge. It acts on another plasma protein, the soluble fibrinogen, which we mentioned earlier, and converts it into insoluble active fibrin threads.
Thrombin converts fibrinogen into fibrin threads. These are the actual mesh builders.
Exactly. Step 5, clot formation. These fibrin threads polymerize and cross-link, forming that dense, sticky network. This mesh traps blood cells and platelets, solidifying into the clot that seals the wound.
Fibrin mesh trap cells forms the clot. Okay.
One more crucial player here, calcium ions. Ka++. Calcium plays a very important role throughout this entire cascade. Many steps require calcium ions to work properly.
So given how critical calcium ions are, if someone had a calcium deficiency, would their blood clotting process be significantly affected? Like, would it be much slower or less efficient, and could that be dangerous?
That's a very insightful question, linking physiology to potential problems. Yes, absolutely. For your exams, it's vital to grasp this. A significant calcium deficiency would indeed severely impair blood clotting. Since Cal++ is essential for multiple steps in the cascade, low levels would slow down the whole process, potentially leading to prolonged or excessive bleeding. It highlighted how crucial even minerals are.
Right. Calcium is indispensable for clotting. Okay, that covers blood and clotting. What about lymph?
Good transition.
Yeah.
Let's talk about lymph, also known as tissue fluid. It's another vital body fluid, working closely with blood. Think about blood flowing through the tiniest blood vessels, the capillaries, in your tissues. The walls are very thin. As blood passes through, Some water and small dissolved substances like nutrients, oxygen, salts filter out of the capillaries into the spaces between the tissue cells.
So fluid leaks out of the capillary.
Yes, a constant, slight leakage.
Yeah.
Larger things, like big proteins and most blood cells, usually stay inside the capillaries because they're too large to pass through easily. This fluid that filters out and surrounds the tissue cells is called interstitial fluid, or tissue fluid.
Interstitial fluid, the fluid between the cells. What's in it?
Its mineral composition is very similar to plasma, since it came from plasma. But, and this is a key difference often asked in exams, it generally has much less protein and far fewer formed elements than blood plasma.
Less protein, fewer cells. What does it do?
It's the crucial go-between. All exchange between the blood in the capillaries and the actual tissue cells happens through this interstitial fluid. Oxygen goes from blood through fluid to cells. Waste goes from cells through fluid to blood.
Ah, the middleman for exchange.
Exactly. Now, this fluid can't just accumulate in the tissues. It needs to be drained away and returned to the circulation. That's the job of the lymphatic system, a network of vessels that collects this interstitial fluid. Once this fluid enters the lymphatic vessels, we call it lymph.
So interstitial fluid becomes lymph when it enters lymphatic vessels.
Precisely. Let's look at lymph's key features and functions. It's a colorless fluid, no RBCs. It contains specialized white blood cells, particularly lymphocytes, making it a major part of the immune system. Lymph nodes, which filter lymph, are packed with these immune cells. Lymph also acts as a carrier for some nutrients and hormones. And critically, it plays a unique role in fat absorption. Fats absorbed from your intestine mostly enter specialized lymphatic capillaries called lacteals in the intestinal villi, rather than going directly into the blood. This fat-rich lymph, called chyle, then travels through the lymphatic system before entering the bloodstream.
So if I'm picturing it right, lymph acts like a very clever middleman. It facilitates all the exchange between the main bloodstream and the individual cells, but it also has its own dedicated immune functions and special transport jobs, like specifically absorbing fats.
That's an excellent analogy. Middleman captures it well. It's the local delivery and waste pickup service for tissues, ensuring smooth communication between blood and cells. Plus, it handles those special tasks like fat absorption and immune surveillance. Really indispensable.
Okay, blood and lymph covered. How does this fluid actually move around the body? The circulatory pathways.
Right. Let's look into broader circulatory pathways. There are two main types found in the animal kingdom: open and closed circulatory systems.
Open and closed, what's the difference?
In an open circulatory system found in arthropods, like insects and mollusks, the heart pumps blood through vessels, but the blood eventually flows out into open spaces or cavities called sinuses. The blood directly bathes the organs in these sinuses. There isn't a continuous closed loop of vessels. It's less controlled.
So the blood just sort of sloshes around the organs?
In a way, yes. In contrast, a closed circulatory system found in annelids, like earthworms, and all chordates, including us, keeps the blood contained. Blood pumped by the heart always circulates within a closed network of blood vessels, arteries, veins, capillaries. It never just spills out into body cavities.
And that's considered better?
Generally, yes. It's considered more advantageous because the flow of blood can be much more precisely regulated and directed to specific tissues based on need. It allows for higher pressure and faster transport, supporting higher metabolic rates.
Makes sense. Now, with invertebrates, how did the heart evolve?
Good question. There's a clear evolutionary trend in vertebrate hearts. All vertebrates have a muscular chambered heart. Fishes have the simplest. A two-chambered heart, one atrium, one ventricle. They have single circulation. The heart pumps deoxygenated blood to the gills. Blood gets oxygenated blood goes to the body. Deoxygenated blood returns to the heart. Blood passes through the heart only once per circuit.
One loop. Okay. What about amphibians and reptiles?
Amphibians and most reptiles, except crocodiles, have a three-chambered heart. Two atria, left and right, but only a single ventricle. Oxygenated blood from lungs, skin, gills enters the left atrium. Deoxygenated blood from the body enters the right atrium. But then they both empty into the single ventricle where some mixing occurs. The ventricle pumps out this mixed blood. This is called incomplete double circulation.
Mixing, so less efficient than it could be.
Right. Then we get to crocodiles, birds, and mammals, including us. We have a four-chambered heart, two atria, and two completely separate ventricles. Oxygenated blood stays on the left side, left atrium to left ventricle. Deoxygenated blood stays on the right side, right atrium to right ventricle. There's no mixing in the ventricles. This allows for two completely separate circuits, one to the lungs, pulmonary, and one to the rest of the body, systemic. This is double circulation.
So the clear evolutionary trend here is a move towards a more efficient and complete separation of oxygenated and deoxygenated blood within the heart itself. And is that related to higher metabolic rates?
No, precisely. You've nailed the core evolutionary driver and the connection. This separation maximizes oxygen delivery efficiency, which is crucial for warm-blooded endothermic animals like birds and mammals with high energy demands. Complete separation supports a more active lifestyle.
Okay, so humans have that efficient four-chambered heart and double circulation. Let's dive into the human system specifically.
Absolutely. The human circulatory system. Components. The muscular four-chambered heart, the network of closed blood vessels, and the blood itself. Let's start with the heart's anatomy. The human heart. It's a mesodermally derived organ located in the thoracic cavity between the lungs, tilted slightly left. Size of a clenched fist. Protection. It's enclosed in a double-walled sac called the pericardium, containing lubricating pericardial fluid to reduce friction during beats.
And the four chambers. 2 atria, 2 ventricles.
Correct. 2 smaller upper atria, receiving chambers, and 2 larger muscular lower ventricles, pumping chambers. They're separated by walls or septa. The thin inner atrial septum separates the atria. The thick-walled interventricular septum separates the ventricles. And the atrioventricular septum separates the atrium and ventricle on each side.
Walls for separation. What about ensuring blood flows the right way? Valves.
Exactly. Valves prevent backflow. Between the right atrium and right ventricle is the tricuspid valve, 3 flaps or cusps. Between the left atrium and left ventricle is the bicuspid valve, also called the mitral valve, 2 cusps.
Tricuspid on the right, bicuspid mitral on the left. Got it. Any others?
Yes, the semilunar valves. One guards the exit from the right ventricle into the pulmonary artery. The other guards the exit from the left ventricle into the aorta. They prevent blood from flowing back into the ventricles after they pump.
So valves ensure one-way traffic. Makes sense. What about the heart muscle itself?
The entire heart wall is made of specialized cardiac muscle. And notice this, the ventricular walls are much thicker than the atrial walls.
Why thicker?
Because they have to pump blood much further and against higher pressure. The right ventricle pumps to the lungs, but the left ventricle has to pump oxygenated blood to the entire rest of the body. It needs much more power.
Higher workload requires stronger walls.
Why?
Now, how does the heart beat on its own? The pacemaker.
Right. This involves the nodal tissue and explains why the heart is myogenic. There's specialized cardiac muscle, the nodal tissue, distributed within the heart. It can generate electrical impulses automatically. Yes. The main player is the sinoatrial node, SAN, located in the upper right corner of the right atrium. The SAN generates action potentials fastest, about 70-75 per minute. It sets the rhythm. That's why it's called the pacemaker. It initiates the heartbeat. Average heart rate, around 72 beats min.
SN is the pacemaker. Where does the impulse go next?
It spreads through the atria, causing them to contract, and reaches the atrioventricular node, AVN, located in the lower left corner of the right atrium near the septum. The AVN picks up the signal, but importantly, it delays it slightly, just for a fraction of a second.
Why the delay?
Crucial. It allows the atria time to finish contracting and empty their blood into the ventricles before the ventricles start to contract. Ensures efficient filling.
Smart design. Then where?
From the AVN, the impulse travels down the atrioventricular bundle, AV bundle, or bundle of His, which runs through the septum between the ventricles. This bundle then divides into right and left branches, which further subdivide into tiny Purkinje fibers spreading throughout the ventricular walls. This network ensures the electrical signal reaches all parts of the ventricular muscle rapidly and simultaneously, causing a coordinated, powerful contraction.
So the heart essentially generates its own electrical impulses, making it myogenic, but the SAN is like the natural leader or the primary conductor that sets the overall pace and rhythm for the entire heart, ensuring everything stays in sync.
Precisely. That's an excellent analogy. The SAN is the internal clock, the conductor. This intrinsic ability to generate its own rhythm without needing nerve signals for every single beat is fundamental. It's quite amazing, isn't it?
It really is. Okay, so that's the electrical system. How does that translate into the actual pumping, the cardiac cycle?
Exactly. Let's walk through the cardiac cycle, the sequence of events in one heartbeat. It involves systole, contraction, and diastole, relaxation of the chambers. Since the heart beats about 72 times a minute, one full cycle takes roughly 0.8 seconds. Let's trace the flow, step by step, slow and steady. Phase 1, joint diastole. We start with all 4 chambers relaxed. Atria and ventricles are in diastole. The AV valves, tricuspid and bicuspid, are open. Blood flows passively from the vena cava into the right atrium and ventricle, and from the pulmonary veins into the left atrium and ventricle. The semilunar valves are closed. About 70% of ventricular filling happens here.
Okay, everyone's relaxed, filling up passively.
Phase 2, atrial systole. Now the sand fires. An electrical impulse spreads across the atria, causing them to contract simultaneously. Atrial systole. This contraction gives a final push, squeezing the remaining blood about 30% from the atria into the ventricles, tops them off.
Atria contract. Finish filling the ventricles.
Phase 3, ventricular systole. The impulse reaches the ventricles via the AVN, bundle of His, and Purkinje fibers. The ventricles contract powerfully. As ventricular pressure rises, it slams the AV valves, tricuspid and bicuspid, shut. This prevents blood from flowing back into the relaxing atria. This valve closure causes the first heart sound.
Lub.
Yeah.
AV valves closing. Okay.
Ventricular pressure keeps rising. When it exceeds the pressure in the pulmonary artery and aorta, it forces the semilunar valves open. Blood is ejected forcefully from the right ventricle into the pulmonary artery and from the left ventricle into the aorta.
Ventricles pump blood out into the arteries.
Phase 4, ventricular diastole. Now the ventricles relax. Ventricular pressure drops rapidly. As soon as ventricular pressure falls below the pressure in the aorta and pulmonary artery, blood starts to flow back slightly, snapping the semilunar valves shut. This closure causes the second heart sound.
Dub.
Dub. Semilunar valves closing. Right. Ventricular pressure continues to fall. When it drops below atrial pressure, the AV valves, tricuspid and bicuspid, are pushed open again by the blood that has accumulated in the atria. Ventricles start filling passively again, and we're back in joint diastole, ready for the sand to fire again and start the next cycle. Lupped up. 0.8 seconds per cycle.
So, to make sure I have this clear for the exam, lub is the sound we hear when the atrioventricular valves, the tricuspid and bicuspid, close, preventing backflow into the atria. And dub is the sound when the semilunar valves close, preventing backflow from the arteries into the ventricles. Is that the essential takeaway for remembering it for NEAT?
Absolutely. You've got it spot on. Lub-dub, the sounds of valve closure. Knowing which valves cause which sound is classic NEAT material. Fundamental. Great job connecting that.
Okay, lub-dub understood. What about how much blood is pumped? Cardiac output.
Good point. Important calculations for exams. First, stroke volume. That's the volume of blood pumped out by each ventricle during one cardiac cycle. It's about 70 millilb in a healthy adult at rest.
70 millilbs per beat, per ventricle. Okay.
Then cardiac output. This is the total volume pumped by each ventricle per minute. Calculated simply. Stroke volume x heart rate. So 70 millbeat x, say 72 beats minute equals 50-40 millimil minute. Roughly 5 liters per minute.
5 liters a minute. That's basically our entire blood volume circulating every minute.
Pretty much. It's incredible. And remember, the body can adjust this. Athletes, for example, can increase their stroke volume and adjust heart rate to achieve much higher cardiac output during exercise. The system is adaptable.
Adaptable. Okay. What about ECG, that pattern on the hospital monitor?
Ah, yes, the electrocardiogram.
Yeah.
ECG. That familiar pick, bip, bip, bip. The machine is an electrocardiograph. The recording is the electrocardiogram. It's a graphical representation of the heart's electrical activity during a cardiac cycle. It's like listening in on the heart's electrical conversation.
How is it recorded? Those pads on the skin?
Yes. For a standard ECG, 3 electrical leads are attached, usually 1 to each wrist and 1 to the left ankle. Sometimes more leads are placed on the chest for a detailed view. These leads detect the tiny electrical changes on the skin surface that result from the heart's activity. The ECG trace shows distinct waves, P, QRS, and T. Each represents a specific electrical event.
P, QRS, T, what do they mean?
The P wave is the first small upward bump. It represents the electrical excitation, the depolarization of the atria. This electrical event triggers atrial contraction, atrial systole.
P wave, atrial depolarization contraction.
Right. Then comes the QRS complex. It's usually the largest, sharpest part. It represents the depolarization of the ventricles. This triggers ventricular contraction. Ventricular systole starts soon after Q.
QRS complex ventricular depolarization contraction. Why is it bigger?
Because the ventricles have much more muscle mass than the atria, so the electrical signal is stronger. Finally, the T wave. It's a broader upward wave after the QRS. It represents ventricular repolarization, the ventricles returning to their resting electrical state, preparing for the next beat. The end of the T wave marks the end of systole.
T wave, ventricular repolarization, relaxation. Okay. How is this useful clinically?
Immensely useful. First, you can calculate the heart rate by counting the QRS complexes over time. More importantly, the shape and timing of these waves are very consistent in healthy individuals. Any deviation, a weird shape, a missing wave, abnormal timing can indicate a problem. Arrhythmias, heart muscle damage, conduction issues. The ECG provides vital clues.
So if I'm understanding this correctly, the ECG basically tells us if the heart's internal electrical wiring system is functioning correctly and in sync, and any abnormality in these waves can point to a potential heart problem or disease. It's like a diagnostic map of its electrical activity.
That's a perfect way to put it. A diagnostic map of the heart's electrical activity. An irregular pattern gives doctors crucial information non-invasively. Your understanding is spot on for the exam.
Great. So we have the heart, the cycle, the electrical activity. What about the pathways again? Double circulation.
Yes, let's revisit double circulation in detail. Remember, blood flows strictly through blood vessels, arteries, veins, capillaries. Quick recap on vessel structure. Arteries and veins have 3 layers. Inner, tunica, intima, smooth endothelium. Middle, tunica media, smooth muscle, elastic fibers thicker in arteries. Outer, tunica externa, connective tissue for support.
Intima, media, externa. Got it. Now the 2 circuits.
Right. 2 distinct circuits originating from the heart. 1, pulmonary circulation. The circuit is for gas exchange. It starts when the right ventricle pumps deoxygenated blood into the pulmonary artery. This artery carries the blood to the lungs. In the lung capillaries, blood releases CO2 and picks up O2. The now oxygenated blood returns from the lungs via the pulmonary veins to the left atrium. That's the pulmonary loop. Heart, lungs at heart.
Right ventricle, pulmonary artery, deoxy, lungs, pulmonary veins, oxy left at atrium.
2, systemic circulation. This circuit supplies the rest of the body. It starts when the powerful left ventricle pumps oxygenated blood into the aorta, the largest artery. The aorta branches into smaller arteries, arterioles, and finally capillaries, delivering O2 and nutrients to all body tissues. In the tissues, blood picks up CO2 and waste products, becoming deoxygenated. This deoxygenated blood is collected by venules, veins, and finally the large vena cavae, which empty it into the right atrium. That's the systemic loop. Heart, body, heart.
Left ventricle, aorta, oxy body tissues, vena cava, deoxy. Atrium. So the pulmonary loop is completely dedicated to getting oxygen into the blood at the lungs, and the systemic loop is all about distributing that oxygenated blood everywhere else in the body and then bringing the deoxygenated blood back to the heart. It's 2 distinct cycles working as one.
Precisely. 2 distinct loops, perfectly synchronized. It ensures highly efficient oxygen delivery and waste removal throughout your entire body, a key feature of mammalian circulation.
Are there any other special circulatory pathways?
Yes, a couple of important ones for NEAT. The hepatic portal system. This is unique. A vein, the hepatic portal vein, collects nutrient-rich blood from the intestine and carries it directly to the liver first before it goes into the main systemic circulation.
Why to the liver first?
For processing. The liver can process absorbed nutrients, like storing glucose, and very importantly, detoxify potentially harmful substances absorbed from the gut before they reach the rest of the body. A crucial checkpoint.
Liver processes the intestinal blood first. Smart. What's the other one?
The coronary system. This is the heart's own dedicated blood supply. The heart muscle is working constantly. It needs its own oxygen and nutrients. Coronary arteries branch off the aorta to supply the heart muscle, and coronary veins collect the deoxygenated blood. Blockages here cause heart attacks.
So the heart pumps blood everywhere, but also needs its own supply via the coronary system.
Okay, now, how is all this activity controlled? Regulation. Right, regulation of cardiac activity. Although the heart is myogenic, its activity is constantly adjusted. There's intrinsic and extrinsic control. Intrinsic regulation is the heart's self-regulation via the nodal tissue, the sand setting the basic rhythm.
The pacemaker controlling itself.
Yes. But then there's extrinsic regulation, modulation from outside systems, primarily the nervous system and hormones. Neural control comes from the autonomic nervous system, ANS, coordinated by a center in the medulla oblongata of the brainstem. The sympathetic nerves, fight or flight, increase heart rate, increase contraction force, and thus increase cardiac output. Gets you ready for action.
Sympathetic speeds it up. What about the opposite?
That's the parasympathetic nerves.
Yeah.
Rest and digest. They decrease heart rate, slow conduction speed, and decrease cardiac output. Calms things down.
Parasympathetic slows it down. Nerves adjust the rate. Hormones, too.
Yes, hormonal control. Adrenal medullary hormones, mainly adrenaline, epinephrine, can also increase cardiac output, similar to the sympathetic system. That's why your heart pounds when you're scared or excited.
So our brain and certain hormones can basically fine-tune and dynamically adjust the heart's natural rhythm and pumping power, increasing or decreasing it based on our body's immediate needs and circumstances, whether we're stressed or relaxed.
Exactly. That's the key takeaway. The heart beats on its own, but its performance is constantly fine-tuned to match what your body needs moment to moment. Incredibly adaptive.
Amazing system. Okay, finally, what can go wrong? Disorders.
Yes. Let's cover some common disorders of the circulatory system, important for exams and general awareness. 1, high blood pressure, hypertension. Very common. Blood pressure is normally around 220/80 millimeter Hg. 120 is systolic, pumping pressure. 80 is diastolic, resting pressure. Hypertension is diagnosed if pressure is consistently 420/90 or higher. It's risky because it strains the heart and blood vessels, increasing risk of heart disease, stroke, kidney damage, often called the silent killer, as it may have no early symptoms.
120/90 or higher is hypertension.
Risky. Okay. 2, coronary artery disease, CAD, also known as atherosclerosis. This affects the coronary arteries supplying the heart muscle, caused by buildup of deposits, calcium, fat, cholesterol, fibrous tissue forming plaques inside the arteries. This narrows the lumen, the opening, reducing blood flow to the heart muscle.
Plaques narrow the heart's own arteries. Dangerous.
Very. Leads to angina and heart attacks. Which brings us to 3, angina, angina pectoris. This is chest pain caused by insufficient oxygen reaching the heart muscle, usually due to CAD. It's a symptom, a warning sign. Can happen at any age, more common in middle-aged elderly. Needs medical attention.
Angina acutes chest pain from lack of oxygen to heart muscle.
Right. And 4, heart failure. This is when the heart isn't pumping blood effectively enough to meet the body's needs. The pump is weakening, sometimes called congestive heart failure, because fluid can back up into the lungs, causing congestion and shortness of breath. Now, crucial distinctions for exams. Heart failure is not the same as cardiac arrest or a heart attack.
Okay, how are they different?
Cardiac arrest is when the heart stops beating completely, an electrical problem. Heart attack, myocardial infarction, is when heart muscle tissue dies due to a sudden blockage in a coronary artery, a plumbing supply problem. Heart failure is the pump becoming inefficient over time.
So, if I understand the distinction clearly for my exam, heart failure means the heart's pumping efficiency isn't enough to meet the body's demands. It's a pump performance issue. It's not that the heart has stopped entirely, like in cardiac arrest, nor is it that a part of the heart muscle has been damaged by a blockage, like in a heart attack.
Precisely. You've nailed those crucial distinctions. Failure, inefficient pump. Arrest, stopped pump, electrical. Attack, damaged pump muscle, blockage. Understanding these is vital.
Got it. That covers the major disorders.
Indeed. So, wrapping up this deep dive, what does it all mean for you? We've seen the vital roles of blood and lymph in transport, defense, balance. We've appreciated the heart's complexity and efficiency as a pump, the elegance of double circulation, the importance of regulation, and, sadly, what happens when things go wrong.
It's a huge amount of interconnected information.
It is. This dive covered everything from tiny platelets to the mighty heart. Remember, visualizing these processes helps immensely. Draw the cardiac cycle. Trace the blood flow. Connect it to reality, my advice. Go over these concepts again. Practice those past year questions specifically on this chapter. Don't just memorize. Understand the why.
Understand, don't just memorize. Okay.
And if doubts come up while revising, reach out. Keep putting in the effort. Consistent work is absolutely the key for NEET and your boards. You're doing great.
Thanks. I feel much clearer on this chapter now.
Excellent. And one final thought to leave you with. Considering how intricate and adaptable this whole circulatory system is, constantly maintaining balance despite challenges, what does that suggest about future medical possibilities? How might we better leverage this understanding to treat or even prevent circulatory diseases down the line? Something to ponder.